everyone. Can you hear me in the back? So I have designed this talk in modules because my understanding is that most of you are astronomers and physicists with very little background in biology. So my philosophy is no matter how much you know about a subject, it is often very helpful to have a simple beginning to reinforce the key points. And so that's what I'm going to begin with, is something that is designed to be understandable to hopefully every single one of you. And then depending upon your level of interest, I'm prepared to go into much more depth on certain topics. Okay, so my hope is that this is interactive, that I'm not just speaking at you. And so please raise your hand if you would like to discuss a point I make or if something I say is unclear. And if you fail to do that, I will prompt you with a question myself as we go, all right? So I understand that this symposium is designed to help you think more critically as astrobiologists or as astrophysicists who are interested in the search for life about how to do that in an informed way. And so I thought I would begin with this iconic photo of Neil Armstrong on the moon because not only is it beautiful, but I think within this photo there is an implicit emphasis and bias that permeates the search for life. And that is, as you see, Armstrong is wearing a spacesuit, and it's because without that spacesuit, he won't be able to exist very long on the moon. And in particular, it's because he needs oxygen in order to breathe. Oxygen has become so part of our awareness of life that you know, I'm sure quite better than I, how it informs the way we look at planetary spectra and assess whether or not a planet has potentially a biosignature. Let me see if I can find a pointer. Is there a pointer somewhere? The bottom one? Okay, great. Yeah. So these type of IR planetary spectra are ones you probably work with all the time, and I need not elaborate on them. My point simply is that when we look at them and when we hear discussions of the search for life on other planets, the emphasis is very much on detecting some signature for oxygen or some form of oxygen in planetary atmospheres, and that's for very good reason. We know this characterizes the Earth. We know this is a key biosignature on our planet today. We don't see it on Venus and Mars, and it's obviously an important thing that could be a biosignature elsewhere. But what I want to emphasize is that had we been looking for oxygen on this planet, we would have missed the first billion years of life on Earth. And I think I want you really to have that sink in. And what would be very interesting for me is to hear how you plan to look for things beyond spectral signatures of oxygen. What other metrics will you be looking at? And we can get back to that in a little while after I say a few more words. All right, so in thinking about going forward in the search for life, I think it's helpful to spend a little time in discussing how we recognize ancient life on this planet, right, our positive control. So our planet is roughly 4.6 billion years old, and the search for life on this planet, I think, can be classified in one of three ways. I think that one can consider the genomic record to be a very valuable record of the history of life. This, without question, provides the most detail on all of the phenomenal enzymes that have ever evolved in any form of life, and we can do comparative genomics to understand their relationships to each other and even to infer more ancestral sequences on a more ancient planet. But what the genomic approach never can do is link any of these signatures, these genetic signatures, to a specific point in time. One can do it in a relative way. One can use molecular clocks, but one can't ever really date anything using the genomic record in an absolute sense. And so for that, one needs other tools. And so we turn either to molecular fossils, which can come in one of two forms, either inorganic or organic fossils. Inorganic can be, any element that's preserved in a mineral that we can measure. We can look at these minerals in different ways. We can assess the type of mineral, its crystal habitat, its shape. We can go in more detail and we can look at its isotopic composition, and we can begin to ask questions whether or not there is a biosignature that could only uniquely have been left by life and not by some abiotic process. In addition, we can look at organic molecules and we can pose similar questions. Is this a molecule that we are unable to come up with any reasonable explanation for its synthesis abiotically? Is this a molecule that really demands a biological process based on its chirality, its isotopic composition, et cetera? And then, of course, we also have more traditional fossils. This one is a beautiful Mammoth Tusk that dates to around 35 to 40,000 years old. This is something I think anyone would agree is a nice fossil. But when you go further back in time, how do you detect fossils unambiguously? Like, would you consider this to be a fossil? No. Well, I actually would potentially argue this might be a fossil. It's not definitively a fossil, but what this is something we call a banded iron formation. And these constitute the world's largest source of iron ore on the planet, and you find them in every continent. And if we date the deposits that we can access, and this is an impartial record because, of course, we only can date things we find, and there are likely many more of these that have not yet been happened upon by intrepid geologists to date. So this is a minimum record of what's out there. But more or less, you see a shape like this, where there's some peak of these oxidized iron ore deposits early in Earth history, around 3.8 billion years ago. Then you see many more of them accumulating as we get to the middle period of Earth history, and then they sort of trail off. And the really important thing to understand is how could they form, and ask critically, are there different processes that led to their deposition that are either abiotic or biological, and if they're biological, what type of life form might have been involved? So first off, what are these things? What are they reflecting? Well, what they're reflecting is the oxidation of ferrous iron. That is the reduced state of iron that is found in seawater that emanates from hydrothermal vents at the bottom of the ocean floor. So if you imagine an ancient Earth, before there was any oxygen around, the seas were filled with ferrous iron coming from this geological source. But then at some point, it got oxidized. And the canonical explanation in geology textbooks used to be that this rusting of the ocean was attributable to the advent of the critical process of oxygenic photosynthesis, which was invented by cyanobacteria over 2.4 billion years ago. And I'm sure you're going to learn more about that from Bob Blankenship this afternoon. However, in 1984, Hyman Hartman at MIT posed the theoretical possibility that there could be another biological mechanism for the oxidation of iron that also was photosynthetic, one that utilized ferrous iron as an electron donor in photosynthesis, and completely in the absence of oxygen could lead to the same type of minerals that we find in these deposits. And so you see an example of a culture of bacteria that we call photoferrotrophs that are able to take light, carbon dioxide, and ferrous iron and grow, and in the process generate this rusty mineral. So how do you know which of these processes was involved in the deposition of these ancient deposits? How can you separate them? That's where it gets really interesting, is to think very critically about what are the specific signatures one would look for that would distinguish those two biological mechanisms from each other, as well as possibly from an abiological mechanism on an early Earth that would have stimulated the oxidation of iron in a different way. Okay, so going back to the timeline, it's important to appreciate that microbial life has really dominated the history of the planet. Animal life is just a very small fraction of the history of life on Earth. And if we think about the type of life that existed on the planet, the part that is really important, I think, for you all in your search for life on other planets is to recognize for at least this first billion years of life's history, in all likelihood, oxygen would not have been detectable in Earth's atmosphere. That detection in the atmosphere is reflected in geochemical records we have to date to around 2.4 billion years ago. And so though oxygenic photosynthesis may have evolved prior to that date, and almost assuredly did, a very big question is how it accumulated in the atmosphere. And I think it's very safe to say there were millions, if not a billion years of Earth history where life was present, yet you never would have detected oxygen. Okay, so what I want to spend some time now in a very simple way is to walk you through common assumptions about metabolisms that really derive from animal life, which I'm going to now describe more generically as macroscopic eukaryotic life. I think it comes from, you know, as usual, we're very self-centered, and so it comes from how we think about our own metabolism. And our metabolism is eating and breathing, so we are going to be enjoying lunch after this, maybe not Chicago pizza, but something else. That's our organic carbon, and we're going to be burning that carbon with oxygen and making waste. So this process that we carry out every time we're making energy is what we call aerobic respiration. And it's nothing more than the combustion of a fuel source by oxygen. And that fuel source I'm giving you in the simplest form as glucose, CH2O. All right? So this reaction is cellular respiration. This happens within our mitochondria. And the output of this is carbon dioxide and water. And I think it's probably no surprise to all of you that this is the great cycle between animals and plants that is often what people think of when they first talk about metabolism and the cycling of elements. So CO2 is fixed back to biomass by taking energy, electrons rather, from water and the energy of light that then gets oxidized to molecular oxygen. And that process is what we call oxygenic photosynthesis. Now, what I just described of aerobic respiration and oxygenic photosynthesis carried out in the mitochondrion and chloroplast, respectively, in animals and plants, come from this part of the tree of life. So how many of you have seen this iconic depiction of the diversity of life? Can you raise your hand? So this is very familiar. What I hope is equally familiar to you, because I think it's really important to realize, is that the vast majority of the organisms on this family tree are microbial, and most life on this tree neither requires nor produces oxygen. And so really where it gets interesting is to understand the modularity of electron transfer reactions and how, given energy sources that can be put together in ways that are energetically favorable, life can do many glorious things. So I'm going to now explain this again in a very general way, and we can get into much more detail if that would be helpful. But the key concept is simple. It's that electron transfer between many different substrates, if it is able to be coupled to ion translocation, can power cells. Okay, so how does this work? So if we go back now and we replace that person in the plant with two generic cell types, we can take this formulation that I just showed you and we can make it a little bit more abstract. So I'm going to call this glucose the electron donor, and the combustion oxygen the electron acceptor. And then what I hope you see is that this glucose, which is the electron donor here, winds up getting oxidized, and it becomes the electron acceptor now for this cell. And then the reverse happens here. Oxygen, which was the electron acceptor for this cell, gets reduced to water, and now this becomes the electron donor for that cell. Okay, so cellular energy is going to derive from the chemical potential difference between these food substrates, between the electron donor and the acceptor. And I'm going to show you in just a moment a cycle very similar to the one I described with animals and plants that's based on cycling of iron rather than oxygen and water. Okay, so again, in the most generic formulation, you can substitute A reductant for the electron donor and B oxidant for the electron acceptor, and then just flip which of these is the oxidized or the reduced species below. So what do I mean by cellular energy derives from the chemical potential difference between food substrates if it's coupled to ion translocation? What I mean by that is that within any biological cell that is using what we call oxidative phosphorylation to power energy generation, namely ATP synthesis, the way this works in the simplest conception of it is simply to understand that there is a pathway mediated by enzymes that are either associated with or integrated into a membrane, and that the transfer of electrons from A reductant to B oxidant, or from B reductant to A oxidant, as the case might be, is achieved concomitantly with the translocation of an ion from one side of the membrane to the other. All right, and so here I'm showing this can be coupled to either proton or sodium translocation. And what this achieves is that electron flow through the membrane essentially charges up the membrane like a battery. And before I go on, I just want to note here, this is meant to depict that this particular electron transfer pathway is only made possible because of activation of a chemical through energy from light. That's photosynthesis. Okay, so once this happens, you wind up getting a charge differential around the membrane, and that can be used to do work. And so this is the famous chemiosmotic theory that was articulated beautifully by Peter Mitchell, for which he won the Nobel Prize. And so it is the dissipation of that chemiosmotic potential gradient around the membrane, which we often call the proton motive force, that allows work to be done, specifically ATP synthesis. So how familiar is this to you? Raise your hand if you've seen this and you understand this really well. Okay, either people are being shy or only half of you have seen this. So I can get into a lot more depth on this in a while if you would like me to do. I'm prepared. But let's continue for those of you who haven't heard this, and then we can come back and go into more depth. Okay, so I want to give you a few examples that will help you appreciate the demonstration that we're going to show in a little bit. So as I said, the cycling of oxygen and water can have can come in many forms, and it can have really quite dramatic consequences for the planet. And so I'm going to give you a few examples, and this is true. We can see this both by looking at ancient rock deposits. We can see this both presently, and I think it's extremely important for thinking about the planet's future, the Earth's future, as well as, of course, your search for life elsewhere. All right, so let me give you a dramatic example just to shake it up. And so I'm going to start with an example of methanogenesis, where we're not going to worry so much about what the specific electron donor is for this process, but focus on the fact that carbon dioxide itself can be an electron acceptor. And when that happens, methane is what gets made. And this was appreciated back at the time of the Declaration of the Independence of the United States was signed at that same year. Alessandro Volta, working in northern Italy in Lake Como. This is the same Volta as in the vault. He discovered many important things. And one of the things he liked to do was go out into Lake Como in a boat and poke around with a stick and collect the bubbles that would come up from the sediments at the bottom of Lake Como. And then he would collect them in a chamber that he put his hand on the top and sealed until he had quite a lot of these bubbles. And then he brought a flame and he removed his hand. And this is an experiment that can be incredibly dramatic. And so I'm going to show you two examples of what happens. Here's the first. So this is a really exciting experiment. It's not for the faint of heart. There is definitely a distinct chance of getting singed, but it's worth it because it's just a really cool way of looking at anaerobic microbiology in your own backyard. And so speaking of your own backyard, when you walk out for lunch, the first thing you're going to walk into is Baxter Pond. And so I'm going to now show you a very quick video of my dear colleague, Jared Ledbetter, doing the Volta Flame experiment with a bunch of Caltech students here a few years ago.
2, 3.
So maybe we could like build this into the next program. All right. So methanogenesis is all around us. You can see it outside in Baxter Pond. It's also super important for us to think about. The cycling of anaerobic substrates is extremely important for us to think about on a planetary scale, both to understand the Earth's past and to understand the Earth's future. And it's really never more important than today, when due to global warming, we're about to have, in the coming century, considerable changes to the northern tundra, where there's a potential for the awakening of organisms that are capable of making methane to start getting much more active. And so a question that's really vital for environmental scientists to be able to answer in a predictive way is what will happen? Is it going to escape to space? Is it going to get retained in our atmosphere? Is it never going to get anywhere because you're just going to evolve an organism to consume it and everything's going to be copacetic? We really don't know, but it's very important for this planet's future to understand better. Okay, so let me give a couple more examples. So now on the respiratory end, instead of CO2, so I was talking about CO2 as the electron acceptor, now I'm going to talk about iron 3 as an electron acceptor, and we'll use generic glucose as the carbon source, and we're going to make CO2 and ferrous iron. And actually, this is where being more specific and aware of what the environmental chemistry is winds up becoming really important for thinking about any microbial process, whether it's on this planet or on another. Because the reality is, on this planet, if we're talking about this process in most habitats, we're not talking about dissolved ferric iron. It just doesn't exist in appreciable concentration at circumneutral pH. The form of iron that's relevant there is a mineral form, such as iron hydroxide, that is a solid. And so how an organism is going to extract energy when it's utilizing a solid for respiration requires certain biological strategies that themselves might impart some type of biosignature one would look for. Something to think about is the importance of, as best you can, framing your quest for a biosignature as informed by knowledge of the detailed geochemistry of the planet that you're searching for, because that will change the game for what life is going to need to do. Okay, so one example I'm going to give you is, in addition to having a mineral form, things can get stuck to minerals and they can be solubilized through microbial activities. For example, in the case of arsenic that gets sorbed onto these oxyhydroxides of iron. All right, so bacteria can utilize both the iron substrate itself and the arsenic as an electron acceptor. And one case where this is happening today here on Earth, where it's very significant in impacting the fate and transport both of arsenic and these minerals, is in Southeast Asia, where due to the structure of the societies there and the local underlying geology and issues of water quality that prevent them from being able to very easily take surface waters for drinking water, they've been needing to drill down into groundwater to access clean water. And this sadly led to tragic consequences because of this microbial reaction. Okay, so what I'm going to show you is a situation where microbes close to people's houses are able to feast on latrine waste. That's where the organic carbon is coming from. Couple the oxidation of that to the reduction of iron minerals, releasing arsenic into their drinking water supply. So the most dramatic example of this happening today is in Bangladesh, where over 5 million people are exposed to arsenic concentrations that are 20 times in excess of the World Health Organization's limit. And this is really a mass poisoning problem on just a very tragic scale, leading to arsenicosis, cancer, due to the ingestion of high concentrations of arsenic into drinking water, potable water supplies. Okay, so here's a situation, a common situation. This is a picture in Bangladesh of someone's home next to a pond. And in the back here is a latrine through which organics are going to be leaching into the groundwater in the vicinity of this house, where they drilled throughout Bangladesh pumps to go down into groundwater to get what they thought was going to be clean water. And tragically, instead, what comes up through that water is arsenic, because it has been mobilized by this anaerobic microbial process. And so what has happened is that people have, from different agencies, government agencies, have gone around and marked in red pumps where they know that the water coming out is dangerously high. And so the good news is that there are ways of remediating this with filtration systems that villagers can be taught to make. And this has been something that people have really played a major role in applying the understanding of microbial metabolism to make a big difference, actually, to public health in this country. And so the situation today is much better than it was even a decade ago. Okay, and so then the final, just dramatic example, getting us back to where I began with that illustration of banded iron formations, is to remind you that the consequence of that type of respiratory process I was just telling you about is not only the fact that you can mobilize things that are stuck to iron minerals, but the fact that the waste products that are made can also be beneficial for organisms. Okay, so here's an example, as I illustrated earlier, where ferrous iron is provided to cells, and it's under totally anoxic conditions, ferrous iron, without any light or without any organisms. It looks clear when you just dissolve it in an aqueous solution. But if you put organisms in and you shine light on them, in the presence of ferrous iron, it forms this lovely oxidized mineral. And this is completely in the absence of any oxygen here in the headspace. What you're looking at here is just a reminder for me to mention that microbes are really versatile. Many microbes are very versatile, and so not only can they use ferrous iron as an electron donor, but many anoxygenic phototrophs, as Bob Blankenship will tell you, can use many different electron donors. And so if you give them hydrogen rather than iron, what you'll wind up seeing is this lovely purple pigment that is a reflection of the photosynthetic reaction chromophores in their membranes that appear purple in visible light. Okay, so this oxidation step, when we're thinking now how to interpret this, is this a biosignature or not? Here's where it's relevant, is that in order to get to these rocks, these are an amalgam of oxidized and also some reduced iron minerals as well as silicate minerals. But ultimately, to get to the oxidized iron, this ferric iron had to come from somewhere. And so this first step of oxidation is the key one to really reflect on whether this is a biological process, such as the one I just showed you, or could that have been achieved abiotically? Or if one is looking at this and you see, is it simply a reflection of an oxidative process that was indirectly biological, in the sense that once oxygenic photosynthesis appeared on Earth, oxygen generated by those organisms could have reacted with this ferrous iron and through a chemical process made ferric. I would argue that's still indirectly a biosignature because the oxygen was biological to begin with. All right, so this is, again, sort of the high-level summary. On this planet, we've had a situation where microbes have dominated the history of life, and in the first billion years of their history, it would have been very unlikely to detect oxygen in the planetary atmosphere. So now I throw it back to you, right? This is why you're here. What are you going to look for? Like, how would you have detected it? Can you conceive of measurements that can be made from space that would get beyond a gas in the atmosphere that could be a putative biosignature? I would love to learn more about that. I don't know if now is the time to have that discussion, but I'm sure you've given it quite a bit of thought and are hoping to come up with measurables. And so that's the gigantic challenge. And so I can keep going, and here's now where it can become modular. I can keep going and talk about, in a more generic sense, ways that might be productive to think about the search for life if we're not looking for oxygen per se. I could also take a much deeper dive into telling you about electron transfer metabolism and bioenergetics. So what would be most valuable for you? Would you prefer me to spend the next 20 minutes or so talking more broadly about biosignatures in a way that's not linked to any particular measurable, but rather planetary chemical disequilibria? Or would you prefer to actually have me go into more depth on electron transfer metabolism? So raise your hand if you'd rather have a discussion on biosignatures and layered communities. Okay, I think that's going to win. All right, so I will proceed. That's what I was guessing, so that's my next slide. Okay, so so let's see. So what is life? Raise your hand if you've heard of this famous book by Erwin Schrödinger. Oh, good. Okay, then I will skip this one. All right, and so what I will just add is that I think there's, at least for me, it helps to think about 3 things life must do, and then try to figure out, once we are clear on these as being really critical aspects of life, how this could translate to a biosignature. So obviously it has to exist. And what I think about as existence in a very generic way is preserving internal homeostasis as distinct from what is outside. That process requires energy, and it also creates a situation where we have thermodynamic disequilibrium with the environment. This is something that I've learned from Ken Nielsen. He's going to elaborate upon this more, and hopefully between the two of us and maybe earlier talks you've heard from Steve Benner and those you're going to hear from Bob, you know, some of this will be valuable to you going forward. Okay, of course, persistence is key. And this today we think about with respect to the ability to replicate, repair, defend, to sense and respond to the environment, various aspects of what that means. And then critically, the ability to co-evolve, to respond, and through the response to change what is then possible, a dynamic situation of evolution. Okay, so this I take straight from a beautiful, thoughtful paper that Ken Nielsen and Pan Conrad wrote, I think in 1999, right, Ken, in the Philosophical Transactions of the Royal Society, which I hope we can provide to the group, because I think it's a beautiful paper, and I think it's very deep actually in what it says, because what it attempts to do is to step back and in a general way reflect on biosignatures without pinning them to any particular elements. And so what I took, and Ken can correct me if I misread, but what I took away from this was the idea that biogeochemical disequilibrium is sort of a powerful biosignature. And one can see this potentially in different ways at different scales. But one way one can look at this is in recognizing that often life at very different scales leaves a predictable layered sequence of metabolites. All right, and so I'm going to show you this at 3 different scales. At the micron scale, here what you're looking at is a biofilm, collection of microbes that are attached to a glass slide here, and nutrients have been flowing past them, and they've been allowed to grow into an assemblage of cells that we sometimes call a biofilm. The scale here is around 150 microns in height, and all that's important to appreciate is that this red zone and this yellow zone have very different levels of oxygen and very different types of energy generating metabolisms operating within them. Now, if we go up in scale here to centimeters, this is going to become more relevant to the demonstration we have prepared for you that I'm going to elaborate on in a little while, the Winogradsky Column. What we see is a beautiful layering of green, of pink, and here down below we see black. I'm going to explain what that is in a moment. But we also can see this layering. I should tell you what you're seeing here, these colors reflect different types of phototrophs, different types of photosynthetic metabolisms, and different anaerobic respiratory metabolisms below. Okay, so the commonality is layering for metabolism, and one can also see this at the scale of meters here in the Black Sea. And this was shown through measuring oxygen at depth in the water column, now over 200 meters is this scale, and other metabolites that are either substrates such as nitrate for anaerobic respiration, or products of denitrification such as nitrite. Here now you see products of ferric iron reduction and manganese reduction. Down here below you see evidence of hydrogen sulfide. And there is a predictable order to what you see, and Ken and I will talk a little bit about why you see the order that you do. But what is really critical is that if we were able to poison any of these systems, kill them, this layering would dissipate. It would go away. Okay, and an assumption inherent in this is that the layering arises from biological activity and reflects the fact that the production and the consumption of metabolizes outpaces diffusion and mixing. Okay, and so therefore this layering is a very nice biosignature. It's catalyzed, and it would defy what you would see if everything were just in normal thermodynamic equilibrium. Okay, so you, again, I'm sure have thought about the biosignature of this planet, water in 3 phases, oxygen, chlorophyll. As I began the talk with, we see this through IR spectra in the famous Galileo study by Carl Sagan, after whom this is named, I gather. We could even detect difference in the reflectance that was attributed to different photosynthetic pigments, depending upon where the Galileo was looking. So the question is, again, are any of these really robust? What does it take to create them? And so let's talk about oxygen and what sets the steady state level. Is this something you've already covered in the session? No? Okay, so there's really only one idea here that I think is sort of a high order idea that's really important to understand, is that seeing oxygen in an atmosphere doesn't tell you that you suddenly, had life invent oxygenic photosynthesis. That could have happened well before you would ever see oxygen in a planetary atmosphere because of this cycling, recall? So in principle, oxygenic photosynthesis could have been invented, but had aerobic respiration been around in perfect balance with it, one would not necessarily expect any accumulation of oxygen anywhere. It could be recycled as quickly as it is generated in theory. And so in equilibrium, there's no net accumulation. What is important to understand is that what we see on a planetary scale is really the reflectance of the maintenance of a steady state that is only put in place because either some of the reductants in the system have escaped, all right? So either we've had some reductant escape so that oxygen can accumulate, or we have had carbon burial in the earth to allow oxygen to build up, so that we have a disequilibrium, and we're getting out of having oxygenic photosynthesis and respiration completely balanced. Okay, so we'll get balanced, but we can balance them at a steady state that must require, at the planetary scale, either escape of reductants or their burial. Okay, so let's take a look at this. If we're looking at the atmosphere of the Earth over time, this is taken from Jim Casting's nice Scientific American article, which is now a couple decades old. What you see is that it is thought that methane rose and then it declined. Oxygen, as I've told you, wasn't present up until around 2.4 in the atmosphere, and then it rose in 2 stages. And CO2 is thought to have had a profile that looks like this. So if we're thinking about methane, right, I showed you that dramatic illustration of methane just outside this building. Why is it that we don't have a lot of methane in the atmosphere? What's keeping it low? And so that gets to the issue of the sink and what is setting the steady state level that we have. And so we know today that in the oceans and elsewhere, there are microorganisms that can oxidize that methane, and they're keeping pace with its genesis such that we don't see any net accumulation. Now, in the case of oxygen, what we understand is that today on Earth, about 50% of the oxygen on the planet, this movie isn't showing, that's a pity. What you would have seen had this started rotating is the global ocean chlorophyll signature from a space satellite. All that really matters is to understand that in the oceans, we have marine phytoplankton that are generating roughly half of the oxygen produced on Earth today. So it's not just occurring in the rainforest that we see on land, but we also have a lot of primary productivity oxygenic photosynthesis that is marine. And so today, many of the organisms that are responsible for oxygenic photosynthesis, they're eukaryotic, only they're microbial eukaryotes. They're phytoplankton, such as marine diatoms that make these gorgeous silica frustules. They also make other types of coatings, such as calcite. And this becomes really relevant when thinking about how oxygen accumulates. Okay, so as I just said, primary production is the fixation of carbon dioxide to biomass. And when we're talking about this as oxygenic photosynthetic process, it's when water is used as the electron donor. Now, as these phytoplankton sink or are grazed upon by zooplankton, which then sink, some of this organic carbon winds up settling, these particles, all the way to the bottom of the ocean floor. And they're not completely respired back, right? This organic carbon does not get converted back entirely to CO2 in water. It's not 100% efficient. And so what happens is that it reaches the bottom of the ocean floor, and over geologic time, some of this reductant is buried and then becomes removed from the system and is inaccessible. And so one really classic example of this that we can see in the Earth's rock record is the accumulation of calcite frustules here in the White Cliffs of Dover. And so these White Cliffs are comprised of nothing more than concentrated amount of calcite from the shells of these beautiful phytoplankton that we call coccolithophores. And so here you can see them in one beautiful example. But I think it just gives a sort of visceral appreciation for the fact that that biological pump is what drove the carbon out of the system and helped allow for the net planetary oxidation leading to the rise of oxygen that we see. Okay, so with that, I think maybe it's time to transition to the Winogradsky column, because this is going to be a demo that will help you appreciate the layering that microbes can establish and the very many different substrates that they can use in order to establish such beautiful layers. Now, it's named a Winogradsky column because the person who, one of the really founding fathers of environmental microbiology is Sergei Winogradsky, this rather severe-looking individual here, who was really quite a genius when it came to being able to go out into nature and make careful observations about the potential energy sources available for life, and then come up with very elegant and very simple experiments in the laboratory to test them. And so he was the one who figured out that all different forms of electron donors could sustain life coupled to CO2 fixation. This is something that includes ferrous iron, reduced forms of nitrogen, you name it. He recognized that the panoply of substrates that in principle could drive growth were really, really vast. And so to illustrate the diversity of substrates that can be used to sustain life, an exercise that environmental microbiology students often do is make a column called a Winogradsky column in his honor. And my student, Jade Livingston, is going to tell you how she did this and is going to walk you through the various Winogradsky columns we have up front. She did some experiments where she wrapped them in foil to block out light, and others where she put them in the windowsill and allowed the sun to get to them. And you can see how that light affected what resulted. All right, but the key thing, I'm going to let Jade explain more of this, but just the bare bones is that the top of, throughout this, there's water. There's air here at the top, and usually we seal this with saran wrap and a rubber band. And here in the base is sediment. And we got our sediment from just outside this building. But over time, if you just if the sediment and settle to the bottom and you put a few other things in there, over time, what you wind up seeing is a layering of colors, and again, in a very predictable way. So green on the top, pink in the middle, and darker down below. And if one were to take a microsensor and profile for compounds of interest, such as oxygen, what you find is that at the top, in the surface waters, they're oxygen replete, and as you would go down at depth, oxygen would become indetectable. And the converse would be true if one were looking for hydrogen sulfide. You would see a lot of hydrogen sulfide at the bottom, and you wouldn't be able to detect any at the top. Okay, and so what I'm going to explain to you now is how the stratification arises from co-selection by light, oxygen, and sulfide. Okay, so just before I get into the details of that, just to sort of hammer home the point, you see this everywhere. And so you can see this layering, the same exact layering on the top, sort of a goldish band. These are diatoms. Then underneath a green layer, cyanobacteria, and under that a purple layer, anoxygenic phototrophs. Here, this is in Yellowstone, a picture from Dave Ward. If one were to go to Guerrero Negro in Baja California and look at the intertidal cyanobacterium mats, this is an image John Grotzinger gave me. Again, you see these beautiful lamina, and if you were to zoom in, you would see all sorts of gorgeous bacteria doing all different types of physiologies. And what's important to realize is that their position is dynamic. And so right here, what you're looking at, this is an organism called Beggiatoa, and it is making a living by oxidizing sulfide as an electron donor and coupling that to CO2 fixation. This is chemoautotrophic metabolism. And the oxidized product of sulfide is elemental sulfur, which forms these beautiful pearl-like objects that you see inside this filamentous this filamentous Beggiatoa. Okay, but these Beggiatoa, they can migrate. So at night, they wind up going up, and during the day, they go down. Okay, and so why are they migrating? What's going on? Well, there are different drivers of this. So abiotic factors that we need to think about is the penetration of light. So this would facilitate, I'm giving this as like a positive driver in that depending upon an organism's possession of certain chromophores, the penetration of light at depth will select nicely for different types of phototrophs that have photosynthetic apparatus to make use of that particular light quality. A negative factor would be the presence of a chemical that's toxic, for example, sulfide. Biological factors, positive, metabolic cooperation. And so while we're probably going to just stick mostly with presenting for the rest of the day to you lessons about metabolism that are driven by equilibrium thermodynamic calculations regarding the free energy to be gained from coupling the oxidation of one electron donor to the reduction of another electron acceptor. What's also really important to know is that that process itself is one that requires, at least in life as we know it, trace elements, vitamins, things that are part of the catalysts that are responsible for driving either an oxidative or a reductive process. And so it's more complicated than just having, you know, substrate A and substrate B. You can have those substrates available, but if you don't also have the trace elements and the vitamins and cofactors that are important to drive the catalysis, then you won't have life, you won't get anywhere. And so from that comes the importance of appreciation of metabolic cooperation and exchange of cofactors and necessities for life between organisms in a community. So it's almost, you could consider an emergent property where in isolation, an organism would not be able to grow no matter how abundant the substrates would be in that environment. So it's a more subtle aspect. And of course, in terms of ecology, one needs to think about competition for resources in terms of resulting in any net steady state profile of organisms that one would see. Okay, so we have to think about predators like viruses or recyclers. How are we going to be able to have a sustainable amount of fuel for a community to exist? All right, so here now just getting a little more specific, and I'm hoping this is going to set Bob up, and then he's going to tell you a lot more about the electron transport chains that underpin these processes. As I have mentioned at the top, what we have is the zone of primary production. Okay, and so that is the ability to take CO2 and with the power of light energy to be able to fix it, taking electrons, extracting electrons from water to drive that fixation to biomass. Okay, so this is actually energetically no small feat when we're using water as the electron donor, because it's a terrible electron donor in terms of its redox potential. It's the worst. And so that's why they need to be at the top of the map, because they need to get the most bang for the buck from the light that is penetrating these mats. At a lower level of light quality in terms of the wavelengths of light that are penetrating, that are at depth going more into the IR rather than the UV, different types of phototrophs that we call either purple sulfur bacteria or green sulfur bacteria. These are also able to use light, but they use different wavelengths of light, and they can operate at different energy levels, because it's actually easier to oxidize something like sulfide than it is to oxidize something like water. And this is entirely predictable based upon their midpoint redox potentials and how one can, from their redox potential, calculate the free energy of this reaction. And I think Ken's going to tell you about that. Right, Ken? Yeah. Okay. So I can tell you about that too, but I don't want to be repetitious. I want to set you up. All right. So here we're having the oxidation of sulfide to elemental sulfur. It could be oxidized also to sulfide. There are many different ways you can combine things. But here, I think the key thing I want to stress is that the sulfide that these organisms are getting has to come from somewhere. And as we go down in depth in these mats, we see this black layer. And what we know is that this black layer is reflecting the presence of sulfide, and underneath that, in this same habitat, the existence of organisms that are taking this organic carbon that's being made by the primary producers above, and they're going to be burning it. They're going to oxidize it by coupling its oxidation to sulfate. And the reason this is something not hard to guess is that sulfate, this particular mat was found in a marsh in a brackish estuary right by the coast, the Atlantic Ocean. This is the Sippewissett salt marsh near Woods Hole. And in seawater, sulfate is really highly abundant, millimolar levels. So this is a very nice electron acceptor to find in a marine or a brackish system. But the point is that the product of this respiration, this anaerobic metabolism, is sulfide, which then diffuses up and becomes the electron donor for these phototrophs above. And so if one were to look under the microscope at them, here's how they would look. This is just in this normal light microscopy. One can see the filamentous cyanobacteria here. This is the layer from the purple sulfur part of the mat. This is the layer from the black layer, the sulfate reducers. If you switch on the UV lamp, what you'll see is that there is characteristic autofluorescence of certain light harvesting pigments at wavelengths that we know we can attribute to those pigments that confirm that these cyanobacteria indeed have the type of photosynthetic machinery that is relevant. And so just now, to get a little bit more detailed, if one were to take a deeper dive into the membrane and look at the structure of the photosynthetic reaction center in some of these cyanobacteria, you might see something that looks like this. This is a mix of lots of different proteins that constitute this amazing molecular machine. And really the only part that I want to emphasize is that for this machine to be able to take water and grab electrons from that water, this occurs in what's called the OEC, the oxygen evolving complex, that's part of this incredible photosynthetic reaction machine. It requires light activation in order for the chemistry to work out. And that light is being captured by a variety of pigments including an important one in the reaction center, which is called chlorophyll A. And so this is the structure here of the key chromophore in chlorophyll A. And if one were to take chlorophyll A and look at its absorption spectra as a function of wavelengths, this is what you would see. This was actually done not just for pure chlorophyll A, this was done for these cyanobacteria more broadly, and so you can see other peaks that correspond to different types of light harvesting pigments. These are called carotenoids. And what I want to emphasize here, though, is that this chlorophyll A peak, one of its major absorption peaks, is around 690 nanometers. But if one were to go down and look at the reaction center machinery and do a similar type of analysis for bacteria chlorophyll A and the carotenoids that we find in purple sulfur bacteria, you see a really different spectra. So here again, just looking here now at the peaks associated with bacterial chlorophyll, these shift over into the IR. And so here's where it becomes very beautiful, where you can take that detailed knowledge of the crystal structure and the biochemistry and the light absorption properties of these pigments, and you can map this to a rationalization of how light selects at depth for particular phototrophs. All right, so shorter wavelengths don't transmit as deeply into a sediment or into a sand like this. Really, it's the longer IR wavelengths that penetrate below a couple of millimeters. And it's not surprising that over time, evolution has adapted such that we see the layering that we do, where the organisms that can use an electron donor coming from below, the sulfide, that's easier to activate, have the photosynthetic machinery that is able to take advantage of these longer IR wavelengths. I wanted to do this in real time for you, but it's kind of hard because of the lights. So I think I'm going to just skip this and end. My point here is that the co-selection is not just by light, but also by sensitivity to toxins. So sulfide, it can be a good thing. It depends on the organism. And if I were to make a plot of the specific growth rate for these different bacteria, what you'd find is the ones deeper down have a faster specific growth rate in the presence of sulfide and can tolerate much higher concentrations than these cyanobacteria on the top. And so in that regard, you have, I've given an explanation of two parameters, both physical, light, and chemical sulfide, that help us understand the layering that we see. So I think I'm going to stop now. Is that good? And I can maybe take questions or turn it over to Jade. What's best to do at this time? Let's look at the columns now. Okay, so Jade, come up and please, you can explain how you did this.
So I'm Jade Livingston. I'm a student in the Newman lab. So we made these Winogradsky columns in March. So these are 4 months old, with the exception being the two tallest ones at the very end. Those we made about 3 weeks ago, just to get a bit of a different look at it. So you all came in here and you saw Baxter Pond, which you saw the nice Volta experiment there. You probably also smelled it. So that's where we got most of our sediments from. We got all of those nice pond sediments. We got the pond water, and then we also got these microbial mats that float around. And so that's algae, it's microbes. It's really great. We've put those up together in buckets. And then we took, yes, am I? Okay. Sorry. Can you hear me? Okay, awesome. Perfect, and I can move around too. So, so the main, oh, what do these buttons do? Actually, wait, what do these buttons do? Is there a pointer?
Back, forward,
Thank you. Okay, so the main thing is the inoculum from the pond. And so that is where we got all of our nice microbes. That's where we got most of what is in all of these. But we also supplemented it with a calcium source, which was eggshells, a sulfur source, which was egg yolks, a cellulose source, which was either paper towels or newspapers, and then another sulfur cellulose source, which we use spinach. And so this is what they looked like 4 months ago. First day we built them. As you can see, with and without light, we hadn't put the foil on yet, but they look exactly the same, just dark mud with some stuff floating in it. And so these are them now. And let me see if I can do this. So I don't know, how do I do this? This one. Can we get this? Can we get this one?
That's it. Okay, and then there was a full screen, right?
Yeah, that one. Okay. So you can see after 4 months, it has developed a lot. You can see the distinctive layering. There's the green cyanobacteria at the top, and then right below that, the purple sulfur bacteria. We also have all of this sediment is floating, which we did not actually, I personally did not expect that, because when you see Winogradsky columns canonically, they're all like there's sediment and there's liquid on top. Does anyone have hypothesis why they might be floating? So, Bubbles, exactly. Any idea what the bubbles might be? Hydrogen, maybe? Methane, yes. So we saw out there, or we saw up here, I guess, that big Volta experiment, we saw the big flame. And so in here, there's a whole bunch of methanogens, and out there, there's a whole bunch of methanogens that are constantly producing methane. And so all that methane is getting caught in the sediment and causing the whole thing to float. Now, this was our plus light experiment. You can see, so this was the side that was facing the window, and then this was the side that wasn't. And so there's a lot more light. There's a lot more growth on this side. But this one, this other one, we had, which we did not put any, it has not seen light since March. And so, yeah, very different. And so, but you can see, tellingly, this one's floating too. And so the methanogenesis is still happening, just not the photosynthesis. Yeah. They're sealed. Yeah, these are sealed. These are sealed. We did several different ways of sealing them. So these ones are sealed with saran wrap. These ones are parafilm and foam and a prayer. Is there pressure? I don't think so. I kind of don't want to. I don't want to test it. I'm pretty sure there isn't, but I'm not 100% sure of any of that. I know this one used to be pressurized because it was. This is the one that I kept in my room, and it exploded. So not ideal. But these ones I think were good. Well, it wasn't sealed. It just spilled over. So, yeah. So we have. We also have these ones that we made more recently. And I don't think I'm going to bother opening it up because it's going to look the exact same as this one. But you can see after just about 3 weeks, I don't know if I can get it in the frame. Can I do that? Can you see it? Ah. Ah. I don't know if you can see it. Can someone else? Yeah. Awesome. So can you hold this bit? Show that bit. So you can already see, and this isn't really doing it justice, but there's already a start to that layering where there's red and green and then dark below that. And this is after only 3 weeks. And so you can totally see how this very quickly develops into really beautiful layering. This is an experiment that you all can do at home if you are brave enough. My first time I did it in Woods Hole, actually, from that same marsh that we were looking at pictures of before. That one also exploded on me, so very got to be careful with it. But you can get really beautiful columns out of it, and it's a really cool thing to put in your windowsill. Yeah. What else should I be adding? I think after this, we're gonna you can come up here, right? You can come up here and look closer. I really recommend it. I don't think this fully did it justice. Some of them are really beautiful. Yeah, thank you so much.
So we would be happy to have a discussion if there's some questions. If you'd rather just come up and talk one-on-one, that's okay too. But I thought I'd ask if there's something or a comment. Seems like Stephen better. Yeah. Yeah. Yeah, I don't have an answer. Yep, that's what I asked. So we are asking again with real enthusiasm and can't wait for you to tell us what the answer is. How are you going to solve this conundrum? What are you going to look for? What's measurable? What are the many different things you can measure? So what kind of layers, so the layers that I was showing you are not layers in an atmosphere. So are you able to detect something outside of an atmosphere? Yeah, back. That's back. Another comment in the back. Yeah. So I'm sorry, I couldn't fully hear. How would you detect anoxygenic phototropes? What's the method you're developing? Uh-huh.
So I'm Nikki Toronto from NASA Ames. But for the amoxygenic phototrophs, because they don't make any gaseous products necessarily that would be detectable in the atmosphere, so you have to look at their pigments. So in particular, the bacterial chlorophylls are characteristic of those. So I've been going out into a variety of different environments and measuring the reflectance spectra of the pigment. So most of you are familiar with the red edge of chlorophyll A. This is, for example, the near-infrared edge of bacterial chlorophyll pigments. So it's building a database of those pigments, and then the next question to ask is, can you really see them on the surface of a planet? Remember, this has to be a global scale phenomenon. It has to be remotely detectable. So with Vicky's students that I'm collaborating with, so they're taking the pigment spectra that I measure and then asking the question, can you really see it through different cloud coverage levels and through different atmospheric compositions? So that's one way that you can attempt to detect a primitive phototroph before the evolution of